Hello, everyone! In this Ask a Scientist video, we'll explore soft-clipped reads in next-generation sequencing, what they are, their origins, their significance, and how to minimize their impact. First, what is a soft-clipped read? It's simply a sequencing read where a portion of the read, either the beginning or the end, or sometimes both, doesn't align perfectly with the reference genome. Let's visualize this by looking at the sequencing read on the right. You'll see that the orange portion doesn't quite align with the reference genome. That's a soft-clipped portion. During initial bioinformatic analysis, reads like this will be marked in the alignment file and flagged for further analysis. Later in the analysis, specialized bioinformatic tools will help determine whether these reads represent true biological variants or technical artifacts that arose during sample processing. Now, let's explore where these partially aligned reads come from. One source is the inherent biological information in the input DNA. Some genomic regions, like low complexity or repetitive areas, are challenging to map precisely, leading to soft-clipped reads. Also, various genomic changes, such as structural variants, insertions, deletions, duplications, inversions, or translocations, can produce these partially aligned reads. Sometimes, sequencing data will contain reads that map to more than one place in the genome, either to a non-contiguous part of the same chromosome or to another chromosome altogether. This type of chimeric read may represent a chromosomal translocation in the input DNA and will be labeled differently than the soft-clipped reads in the alignment files. Another key source of soft-clipped reads is artifacts from sample preparation, specifically during library prep, due to technical issues. Before we dive into that, let's see how we measure their presence. To show how different library prep methods can affect the number of soft-clipped reads, we'll use data from one high-quality human genomic DNA sample prepared using 3 different library prep workflows. First, we'll look at data from DNA prepared using mechanical fragmentation prior to library prep. This method, which uses sound waves to break up the DNA, is the gold standard for minimizing soft clipping artifacts because it's random and unbiased. The gray line on this chart shows the number and lengths of soft-clipped reads from mechanically fragmented DNA. While mechanical fragmentation can be excellent for reducing artifacts, enzymatic methods are often faster and require less starting material. However, enzymatic methods can sometimes increase soft-clipped reads and other artifacts. The orange line shows the distribution from the same DNA sample fragmented using a transposon-based enzymatic method that simultaneously cuts the DNA and adds sequencing adapters. This method is more prone to adding bias, often cutting DNA near inverted repeats and leaving overhangs. These features contribute to the larger number of soft-clipped reads you see in this data. Finally, let's consider a high-quality endonuclease-based enzymatic fragmentation solution. The green line shows a pattern much closer to mechanical fragmentation, producing significantly fewer soft-clipped reads than transposon-based methods, though still slightly more than mechanical fragmentation. This highlights that different library prep kits vary in performance, so comparing workflows before a large experiment is wise. Now, let's revisit how soft-clipped reads emerge during library preparation. As I mentioned earlier, some regions of the genome contain naturally occurring inverted repeats, represented here in pink. In the case of soft-clipped reads, part of a sequencing read that originates from these regions can be incorrectly assigned to the opposite strand of genomic DNA during analysis. The blue arrows represent a soft-clipped read that aligns to both the top strand and bottom strand. Let's see how this can happen. A few years ago, Ohio State University scientists studied the origin of enzymatic fragmentation artifacts. The next slides are based on figures from their published paper, cited on screen. One hypothesis is that enzymatic activity during library preparation exposes these inverted repeats as single strands. This can enable intramolecular binding during library preparation, creating a stem loop structure that is susceptible to breakage. Then, during end repair and PCR, DNA molecules can be created that combine material from both of the original strands into a single strand. When sequenced, this can result in partially aligned reads. So, how can you tell if soft-clipped reads are artifacts or if they represent real biological information? Fortunately, free bioinformatics tools exist to help identify artifactual soft-clipped reads. The Gregory et al. Paper cited on the screen describes a tool called FADE, which stands for Fragmentation Artifact Detection and Elimination, which the developers freely share. Some research groups also develop their own specialized tools. There is one more common source of soft-clipped reads: errors during the post-sequencing data analysis. For example, base calling errors made by the sequencer or during bioinformatic analysis can cause soft clipping. Also, if NGS adapter sequences aren't fully removed, you can get unaligned bases in some reads. Now, what's the overall impact of soft-clipped reads on your NGS data's reliability? Fortunately, the occurrence of soft-clipped reads that are not identified during analysis is rare. Fewer than 1% of artifactual soft-clipped reads are mistakenly reported as variants or false positives. Soft-clipped reads can have a greater impact on rare variant discovery or detection than on whole genome sequencing or whole exome sequencing for germline variant analysis. Finally, sequencing data from target enrichment workflows using small panels are more likely to contain soft-clipped reads because any artifacts matching the panel's target regions will be enriched. Now, let's recap. What factors can increase the occurrence of soft-clipped reads, and how can you mitigate their impact? First, while enzymatic fragmentation workflows are often simpler, faster, and require less input DNA, they can lead to higher numbers of artifacts that form during library prep. Second, soft-clipped reads are more common in regions with naturally occurring inverted repeats or other highly repetitive sequences. Finally, target enrichment, especially for smaller regions, can concentrate soft-clipped reads in your raw data. To reduce the likelihood of soft-clipped reads and to mitigate their impact on your results, take the time to consider the trade-offs associated with the different types of library prep workflows and understand which is the best fit for your sample type, sample availability, and laboratory setup. Before embarking on a large-scale experiment, test a representative sample to see how it performs in different workflows. And finally, using the right analysis software can help identify artifactual reads, preventing false variant calls. We hope this video helped you understand soft-clipped reads in DNA sequencing. For more short videos on NGS, target enrichment, or RNA sequencing, visit the Ask a Scientist link on your screen. Thanks for watching!